In the last tutorial, we have prepared our protein, we have prepared our ligand. Now in this tutorial, we will dock them. But before starting with the tutorial, we will make some arrangements. We will copy all these PDBQT files of the ligand and paste them in the scripts folder. At the same time, we will also paste our prepared protein file in the same scripts folder. Next, we will bring our vina.exe file from the Scripps Research Institute folder, which is present in our C drive, where we have originally downloaded our MGL tools. So we will go to local C drive, then program files 86. Here you will find the Scripps Research Institute folder. In the Vina folder, you will get the vina.exe executable file. We will copy that and paste it in our scripts folder. Now we have brought our prepared protein, prepared ligands, as well as the executable file in the scripts folder. Now we will provide the search space for the docking program, where the vina.exe will find the best docking site on the protein and dock our ligand there. For that, we will open our Autodock Tools software and make a grid box. So let's open our Autodock Tools software. Again, we will go to File, go to Preference, and we will set our working directory. We will paste the path to our working directory, that is the scripts folder, in the startup directory icon box or text box. We will copy the path from the top ribbon and we will paste it here, and we will click on set, and then we will dismiss this dialog box. Now to open our macromolecule, we will go to grid, macromolecule, and click on open. So here is our prepared proteins PDBQT file. We will open it. We'll actually, the Autodock Tools is asking us to add extra charges that are the gas trigger charges on the protein, or preserve the previous charges. So we will preserve the previous charges and click on no. Now we will draw a grid box that will uncover the complete protein. This grid box will tell our vina.exe file the required search space, where it will search for the best docking site for our ligands. To draw the grid box, we will go to grid, we will click on grid box, and here you can see the grid box has appeared. Now we will enlarge the grid box so that it can cover the entire protein. Save the computational effort. We will ignore this terminal amino acid chain, and we will include the main chain protein only. So we can enlarge our grid box by moving these wheels. The scroll button on the top that are present under the number of points in X dimension, such scroll bars will increase the size of the complete box, whereas the scroll buttons that are present at the bottom, that is under the heading center grid box, these scroll buttons are used to move the box or translate the box along the X, Y, and Z axis. Therefore, these amino acids are out of the box, so I need to shift the box to that side of the protein. The green surface I need to move. Therefore, I will use the green color scroll box that is present at the bottom of the grid options dialog box, so that I can include those amino acids in my protein structure. Oh, sorry, in the grid box. But as you can see, as we are moving to that side, these amino acids are left behind. So we will do one thing, we will center the box exactly, and we will increase the spacing of the box. This will enlarge the box. This will enlarge the box in all three axes like this. And now we have included the complete main chain protein uh, in the box. We have covered all the main chain amino acids in the box, and we have left these terminal amino acids. But as you can see that we can increase the box dimension, we can include some of these amino acids, but right now we will keep them away to save our computational effort and time. Now we will save these dimensions in a particular file, which we will name it as grid dimension file, or the grid file as short name. So we will go to file and we will click on output grid dimension file. Here we will name the file as grid under grid.txt. Note that it is quite important to provide the .txt extension, otherwise the Autodoc program will make it as a plain text file but without any extension. So click on save, and we will verify whether our Autodoc program has made our desired grid.txt file in the scripts folder. So it has made our grid file, which consists of the name of our protein, then the spacing we have used, then the x, y, and z coordinate of our box, and we have the x, y, and z coordinate of our center of the box. Now that we have provided the search space to our for our bina.exe program to search for, now we will write a configuration file. For this, we will go to Notepad, and here we will write 6 fields that we need to specify, and these 6 fields will be read by the vina.exe program and generate our docking output. So the first field we need to specify is the receptor name. Therefore, receptor equal to. Here we will specify the receptor PWQT file. That is the protein PWQT file. Next, we need to specify the ligand file. Next, we need to specify the dimensions of our uh, grid box. That is the box XYZ coordinate as well as the box's center XYZ coordinate. That is the XYZ coordinate of the center of the box. Therefore, first we will provide the XYZ coordinate of the center of our grid box. This is the X coordinate, and similarly we will write the X, Y, and Z coordinate. Next, we will provide the X, Y, and Z coordinate of the box itself. Next, we will provide the output file location where we want to save our output. So, before writing the path to our output file, we will go to the results folder. Here we will make two subfolders. We will name one subfolder as output out files and another subfolder as log files. In the out files, we will save our output files, which will contain different poses of our ligand that will be docked with the receptor molecule. Whereas the output that would be displayed on the terminal will be stored in a special text file, which we call as log files, and we will store these log files in the log files folder. These log files will hold the binding affinity scores as well as the RMSD values, and these binding scores will be used to compare the docking performance of different ligand with our protein molecule. And next, we will also provide the part 2, the log file. Now, the receptor file name we need to provide. So, our receptor file name is 1m17.pdbqt. So, we will specify that name 7.pdbqt. Next, we will provide the ligand name. I suggest to copy the ligand name directly rather than typing it out. Copy, and here we will paste it. And do not forget to add extension .pdbqt. Avoid making spelling mistakes, as this will cause errors while running the actual docking program. Next, we will provide the center XYZ coordinates here. The center XYZ coordinate is the last line, which will provide one coordinate at a time in the center X, center Y, and center Z rows. So here 19.295 will be the X coordinate of the center of the box. And similarly 6.14 will be the Y, and 59.395 will be the Z coordinate. So I suggest you copy and paste rather than typing it out. Next, we will provide the X, Y, and Z coordinate of the box itself. Now we will write the path where our output file need to be saved. Our output file need to be saved in the results folder, which is present outside the scripts folder. Therefore, we need to tell the vina.exe program that we need to first go out of our scripts program. And to go out of our scripts program, we will type the two dots and followed by a /. This combination of characters will take us outside the script folder. Now the vina.exe program can see that it is already outside the script folder, and it can see the data, images, results, and scripts folder. Now we want to enter into the results folder. Therefore, we write the name of our results folder, results, and /. The results and / will take the vina.exe program into the results folder. In the results folder, we want to save our out files in the out files folder. We will type the name of that folder, out files, and /. The / will take us inside the out files folder. Here we need to save the out file with a specific name, and that name we will enter here, lig_1.pdbqt. Note that wherever you write the file extensions during writing the configuration file, all the files are in pdbqt format. We will provide the same path, but we will save the log files in the log files folder. Therefore, in results folder, therefore inside the result folder, we will be writing the out that is the log files in the log files folder. Therefore, we will change here out files to log files. Rest the path remains the same, and the name of the file will have the extension .log. Now we will save this file with the name config underscore one dot txt file. Now this configuration file in short tells the vina.exe program to dock the receptor, that is 1M17, with my first ligand, that is ligand 1, which is citric acid. And this is the search space. You want to search for best docking site and output the results in the out files folder, and write the binding scores in a log file where you save it, where the log file is saved at this particular location. Now we will make another log configuration files uh, which will hold the information about docking of 1M17 receptor with ligand 2, and the name of the ligand will be different. Also, we need to change the output and log file name as well. So we will copy this configuration file, copy, and we will paste it in new notepad file. Now we will change the ligand name, that is lig2. We will paste it directly so that we will minimize any errors due to a name mismatch. We have pasted the name of the ligand 2 in front of the ligand tag. Now we will change the output file name as well, that is lig2 and log2. We will save this file as config2 file. Similarly, you have to make more 3 configuration file, which will the only difference between those file will be the ligand name will be different, as well as the output and the log file names will be different. As you can see here, we have 5 configuration file. We have the prepared protein, we have the prepared ligands, and we have the Vina executable files. Note that you have to place all these files along with the Vina executable in the same folder while running the vina.exe script. In that way, you do not have to provide the path independently to all the input files. And now we will run the main docking command. So go to a ribbon tab, so go to the ribbon and type cmd to launch the Windows command prompt. Here we will list down the file with the dir command. So here we are in the right folder. Now we can execute our main command. The simple command for one protein and one ligand docking uh, is provided like this. So vina.exe dash dash config, and dash dash config is the tag where we provide the configuration file to the vina.exe program. So for only one protein and one ligand, we will have a single configuration file. For example, config underscore 1.txt, and we will repeat the docking program for 24 times. For that, for the depth of 24, uh, for the depth of 24 operations, and we can specify the depth of operation with the dash dash exhaustive tag, exhaustiveness tag, exhaustiveness equal 24. So this is the single program or the main command line that you have to write for docking a single protein with the single ligand. But in our case we have 5 configuration file, so we need to write this same line 5 times. But this repetitive operation can be optimized by just writing a for loop. So we don't have to write the same line 5 times. For writing a for loop, we will write for and %f. First, I will give you the complete command and then explain what the command means. For %f in, we will write a parenthesis here and specify the wildcard dot txt. Here it is underscore and do, and the main command vina.exe dash dash config %f dash dash exhaustiveness equal 24. Here I have written a for loop, and inside the for loop I have specified our command which we will use for a single protein and single docking case, single protein and single ligand docking case. So the for loop starts with the keyword for, and here we have specified one container %f. %f is just like a container which holds different files when the loop proceeds. Then we have just specified a bucket, for instance, and that bucket will hold our all the configuration files. And how we have filled the bucket? So we have, as you can see, config1.txt, config2.txt, config3.txt. You can see that all these 5 files have same name except the number is changing. In the first one there is 1, in the second there is 2, in the third there is 3, and so on. That means the first part of the file and the next part of the file after the number, that means the part of the file before the number and the part of the file name after the number, is shared by all the files. Therefore this constant character or the constants in the file name we have written here, and the variable part, that is the number, we have replaced it with star. Now the star is holding the variable part and the constant part we have written out. What this actually means is that this star will be replaced by number whenever the loop proceeds. For instance, in the first run of the for loop, here the config1.txt, that means the star will be replaced by the 1, and this config underscore 1.txt will be stored in this %f container, and this %f container will then run this command. That means vina.exe and the config file will be specified, and this %f will be replaced by the contents. That means the %f is holding config underscore 1.txt, and that %f here will be replaced by this config underscore 1.txt name. And so the whole command will look like the one I just showed you. So vina.exe dash dash config config1.txt and exhaustiveness equal 24, and the docking program will be run for our first protein and ligand, first receptor and ligand, or first protein and ligand. Now after the program has run for our first pair, the config underscore star.txt will be replaced by config2.txt, which will be stored in %f, and this %f will be replaced uh, in the main body of our for loop. Now the vina command for the next step will be vina.exe dash dash config config underscore 2.txt, and exhaustiveness will be 24. Again, the docking program will be run for our second pair, and this operation will take place again and again until the vina underscore star.txt ends. Therefore, this is the simple for loop that saves our time for writing the same command 5 times. Now we will click on enter to execute the program. Whenever we need Whenever we provide a big grid box, this analyzing of binding site takes more time, and when the grid box is small, this step takes very less time. Moreover, the docking program that will be run after this analysis of binding site will also take more time when the grid box is larger. So this will take more time in our case as we have larger grid box. Now as you can see, the first command has been executed and it has written the output. And here the second command is executed, where you can see the config.txt file has been provided as the input to the vina.exe program. We can see our outputs in the results folder, and here you can see the lig1 log file has been prepared. You can open it, and here you can see the binding affinity scores that are available, and out file has also been prepared. So we will wait for all the programs to get executed and all the log files and the output files to be outputted in the results folder. In the results.out files folder, we can see that all the output has been written in the form of pdbqt file, and the binding affinities have been written in the log files. So for the first ligand, the binding affinity was minus 5.7. For the second, it was minus 8.8. For the third, it was minus 8.6, comparable to the second. Uh, the binding affinity for the ligand 4 was quite less, minus 4.9. And for the fifth ligand, it was minus 7.3. So we can conclude that the ligand 2 was binding more efficiently as compared to other ligands to our receptor. So now we will open our ligand 2 in PyMol and see where it has actually bind to our receptor. First, we will focus on the right side of our PyMol window. Here you can see uh, that the ligand has been listed, and in the same frame uh, you can see the global frames dialog box has appeared. In the global frames dialog box, you can see there are 9 poses that are loaded, and this is the first pose that is being presented on the molecular viewer window. You can use the keyboard's arrow key button to move from one frame to another frame. So by clicking the single arrow key to the right, this is the second pose, this is the third pose, this is the fourth, this is the fifth, sixth, seventh, eighth, and ninth frame. But from our log file, we can see that the first pose of our ellagic acid has obtained a good binding affinity. Moreover, the RMSD values are close to 0, which is a good docking result. Next, we will see where our ligand has binded over to the protein structure. So we will go to file and load our receptor molecule. As we zoom in our protein ligand complex, here you can see that the ligand exactly overlaps with the natural ligand that is present in our protein. This means the protein's main binding site was occupied by our ligand, which indicates the positive docking result. If we move to other poses of the ligand, you can see that this ligand, all poses are present in the active site of our protein. That means our docking has been properly carried out without an error. If the ligand was present other than the binding site of our protein, then we need to repeat our docking analysis again, because the docking result we would have obtained would be inaccurate. But in this case, our ellagic acid occupies the same position as the protein's natural ligand is uh, have obtained or placed. That means our ellagic acid has occupied the active site of our protein. So in this way, you could confirm your docking result by comparing our docked ligand with the crystal structure of our protein. But now we have confirmed that our docking results are correct. We will remove the crystal structure of protein and visualize with our prepared protein structure. So in the right hand side, you can see the three tabs are open, that is all. Then our ligand is loaded, and then our crystal structure of our protein is loaded. To remove the crystal structure of protein, you can see 1M17 tag or the molecule is present. So in the same line, we will go to action button and we'll go down and we will select delete object button. In this way, your only protein molecule is removed while retaining the ligand molecule. So this is our docked complex that we are visualizing. Here also different poses of ligand can be visualized. Now we want to know which amino acids are interacting with our ligand molecule. For that, we will again go to the right hand side, and here you can again see two molecules are loaded, that is ligand 2 and our receptor molecule. In the receptor molecule, in front of the receptor molecule 1M17 tag, you can see 5 action buttons or 5 buttons. The A stands for the action button, S stands for the show button, H stands for hide, L for label, and C for coloring of your receptor molecule. We need to visualize what bonds our protein molecule is forming with our ligand molecule. For that, we will go to the action, that is A button of our 1M17 protein. Once you click on the A button, you can see the title, that is it is an action button. Go to the find option, find a drop down list. Another drop down list will appear where you can see polar contacts. Go to the polar contacts drop down list and all the way to any atoms. Here you can see yellow lines appeared that starts from the ligand molecule and ends at the amino acids which are not yet seen because the protein structure is in ribbon representation or the cartoon representation. So to see the amino acids that are actually making hydrogen bonds with our ligand molecule, we will close the ribbon representation or the cartoon representation of our receptor, and we will represent the protein molecule in sticks format. To do this, we will again go to 1M17 on the right hand side and we will go to the H button. Click on the H button and click on cartoon. This will hide our cartoon representation of protein. Now we will show our protein in stick representation. For this, again go to the action buttons and in this case click on show action button. and then click on sticks. Here you can see our complete protein structure is now represented as single amino acids in stick representation. Now we will zoom in and manually select the amino acids that are making uh, hydrogen bond with our ligand molecule along with my ligand. Now again focus on the right hand side of our window. Here you can see another selection being added, and the name of this tag is SELE, which stores all the amino acids as well as the ligand we have just selected. We will rename this object by going to action button. Click on a action of the selection uh, button or the selection tag or the SELE tag that have just been created. Click on A and go to the second option, which is called rename selection. Click on that. On the left hand side you can see, here on the left hand side you can see rename SELE to SELE. Here you can specify the name you want to give to the tag on the right hand side. So I will give the name as interacting residues. Interacting residues, and I will hit enter. Now again on the right hand side you can see that the SELE name has been replaced by the name that you have just provided. Next, we want to hide all the other representation, and we want to keep just the ligand and the interacting residue representation. To visualize the interacting amino acid more clearly. Now our 1M17 protein stick representation are hidden. Now we will go to interacting residues and click on S. And in the S drop down, click on sticks. Here we will get a clean interaction of the amino acid of the proteins that are actually interacting and forming hydrogen bond with our ellagic acid, that is our ligand. Now we want to add labels to our moieties. For this we will again go to interacting residues and go to L, which is label, and in the labels we will select that is we will go to L, that is label, drop down, click on residues. Here you can see the labels have been added to our amino acids, but these labels are very small in size and overlaps with our stick representation. We need to move them. In order to move them, we will go to mouse button and we will first change the mouse 3-button mouse modes. Right now the mouse mode is the viewing mouse mode, which you can see on the left hand side bottom ribbon. Here you can see the 3D mouse button is set on viewing mode. We need to bring it to the editing mode. For this, just select on 3-button editing. Now go to the labels and on the keyboard press the control key. Upon pressing the control key, now click on the label and just drag the label where you want to place it. Again, press the control key on the keyboard, click on the label and move the label. Sometimes two labels arise, just place them on one another to hide one extra label. We will also bring this label to the more visible area. Now our amino acids labels can be seen, but the label on the ligand is not visible. In this case, we will provide a custom label to our ligand. For that, again change the mouse mode to viewing, and on the top left corner, that is below the file tab, you can see the residues option being present. Click on it, it will open a drop down list. Click on atoms and click anywhere on the ligand. For example, I will click here. This will select a single atom that is present on the ligand. Again, on the right hand side, you can see SELE name tag has been formed. We will again go to action button and we will give it one meaningful name. For example, I will name it as ligand. Hit enter. You can see the tag is now changed to ligand. Now come to the command line of the PyMol. At the extreme bottom of your uh, PyMol window, type the following command: label, LABEL space, and we will uh, type the name of our selection, that is ligand, comma, and put the inverted commas or the quotation mark. Inside the quotation mark, write the name of the ligand. Here I will label my ligand as ellagic acid. Ellagic space acid. Hit enter. In the molecular viewer window, up where we have selected our atom, you can see that the label has been added. Now we will move this label again by changing the mouse mode. We have changed the mouse mode to editing again. Now press the control key and click on the label and just drag the label where you want. Now as you can see, everything is set. We can visualize the hydrogen bonding in yellow, interacting amino acids in blue color, and our ligand in green color along with their respective labels. But one last thing I need to do is I need to increase the size of the label to improve its visibility. To do this, we will go to the settings, then we will go to label, then we'll go to size, and we will increase the size to 24 points. This gives us the clear 2D representation as well as the 3D representation of the interacting amino acids of the protein with our ligand. In this way, you can visualize the interaction of your ligand with your amino acids present in your protein's active site. Moreover, you can also visualize your protein in some different manner. For example, you can also visualize it in surface representation. This I can show you. So we will reinitialize it. Again, open our protein and our ligand molecule. You will get such representation. Do not worry. Just just use the scroll button which is present on your mouse. We will represent this protein which is currently in the ribbon representation, or you can say cartoon representation, in a surface representation, and visualize where in this surface representation our molecule is fitting. For this, we will again go to the right hand side. We'll go to again S, that is show button, and we will click on surface representation. We will change the color of this surface representation from this green color to one monochromatic color. For this, again go to right hand side, click on C, which stands for color, and I will visualize this protein in for example, some different color like the wheat color, for instance. So here you can see our ligand is present in this binding pocket. Very beautiful representation, and such images you can put it in your docking analysis results. So in this way we have docked multiple ligands with the same protein just using a single command, that was of the for loop. So and we have outputted the complex and visualized it in a PyMol. We have visualized the hydrogen bonding or the polar interaction, as well as the binding site visualization where our ligand fits in the protein's binding site. Moreover, we have also visualized whether our docking results are valid or not. So that's it with this tutorial. And if you like this tutorial, please do subscribe to our channel, and for more such tutorial on bioinformatics, please subscribe to our channel. In the next tutorial, I am basically planning to do tutorial on again on protein ligand docking, but with explicit waters. So stay tuned for that tutorial, and thank you again for watching this tutorial with patience. If you have any doubt, please ping me in the comment section. I will get back to you as soon as possible. So thank you.